Today I'm going to be talking about two separate systems. The CRISPR system that bacteria use and is an immune system, and the CRISPR-Cas9 system that we have hijacked from this CRISPR system to use for other organisms, for our food, for plants, and for even humans. So basically all CRISPR is it stands for clustered, regularly interspaced, short palindromic repeats. And that's a lot, but basically all that means is that we've got two parts. And so in a bacteria, and this is their DNA, we have the interspaced segments of palindromic repeats. So this is what those repeats are. And what palindromic means is that they're the same if you read it forwards or backwards. So this is going to contain the same base pairs information repeated between intervals. And so between these intervals, we've got this spacer DNA. And what researchers found was that in 1987 was that this spacer DNA contained matching viral DNA specifically from bacteriophages. And so this puzzled researchers to find that basically this spacer DNA was a chemical snapshot of foreign pathogens coming in and trying to inject their DNA into the bacterial genome. And so basically how the system works is that we have a bacteriophage, and it's going to insert its DNA into the bacteria. Right now I just have E. coli because that's where they found this system. So it inserts its viral DNA. And so what happens is that this CRISPR locus has these Cas genes, and these Cas genes create proteins that create a nuclease that will go and it will cut specific target genes, and then we have the helicase that's going to just unzip and clip more DNA. And so these Cas genes are going to go and they're going to attack this viral DNA. And they're going to cut it, and they're going to strip it, and they're going to make a single strand of RNA. And then this Cas protein and this RNA, they're going to combine. And they're basically going to be a scout for the bacteria, for the bacterial genome. And so basically, first time that this pathogen is introduced, it's basically like a warning. The second time, it's going to have that information with the Cas genes, and it's going to have the strand of DNA. It's going to go and it's going to attack it, cut it, and kill this viral DNA from infecting it into the genome. So basically, it's an immune system, just like eukaryotes have, just on a smaller scale. And this information is stored in between these segments in the spacer DNA. Okay, so this was discovered in 1987, and so researchers were like, why can't we use this system on our own? Our food, plants, animals, and then humans. And that's where the CRISPR-Cas9 system comes in. And it is basically using this CRISPR system that's naturally found in bacteria. So with the CRISPR-Cas9 system, we've got three components. Just like in the bacteria, we've got the Cas9 protein. This Cas9 protein is it's got the helicase and the nuclease, which means it's gonna go and it's gonna cut. It's gonna cut your target gene. It's got the guide RNA, which is basically a blueprint to say where to go on a specific strand of DNA. And then it's going to have the host RNA. So, and that host RNA is basically going to be the corrected gene. So, for instance, say this is a a human DNA strand. So this Cas9, this guide RNA, and this host RNA are all in this mix, this complex. They're going to go and they're going to attack the specific targeted gene that is programmed into the mix. They're going to insert it into this target cell, and the Cas9 is going to do exactly what it did in the bacteria. It's going to cut that specific segment of DNA. The guide RNA is just telling it exactly where to go, acting like a primer, showing the nuclease where to go. And so what we've got now is we've got this, this DNA has been cut. And so the cell's going to try and repair it, like the cell, and it's going to try, but most likely it's going to fail. It's going to either turn the gene off completely, or it's going to create a mutation, which is not what we're going for in this treatment. And that's where this host RNA comes in. It's going to, this is going to take it, it's going to show us where we're going, cut it out, and then this is going to be the corrected form. And so this host RNA is going to match up, and it will complete the strand. So we have taken out this mutated segment of DNA, replaced it with the correct base pairs to line up, and so now we have formed a corrected strand of DNA. And this is used in various genetic treatments, cystic fibrosis, that's going to go in, it's going to target that CFTR gene, and it's going to replace it with the corrected copy of the mutated DNA. It's going to work with Huntington's disease, sickle cell, cancer, just various genetic disorders, and this is an awesome tool because it is non-invasive, it is cheap, it is simple. There are kits you can use to work with this stuff, and all was found from our good friend E. coli, and it's a natural process, and it is very important. It's a wide open field. It was just, this CRISPR Cas9 system was just founded in 2012, so it's got a lot of promise for the future of medicine.